Hello and welcome to part 3 of the 3.3 Meiosis series. So, what we're going to focus on next is this statement, drawing the diagrams to show the stages of meiosis resulting in the formation of 4 haploid cells. Remember, haploid is half the number of chromosomes and half the number or amount of genetic information. Okay. So here we are in meiosis 1, and remember meiosis 1 starts with these replicated chromosomes. So you've got the X-shaped replicated chromosomes, right? Okay, and this is in prophase 1, right? And so the chromosomes are just kind of floating around in prophase 1, right? And they're also doing that thing called what? Where the genetic information is exchanged and transferred. They're doing something called crossing over, right? It increases genetic variation. Crossing over occurs in prophase 1. Just going to write P1 right there. Okay. And then we have, and also you can see that the nucleus has dissolved and is no longer there. Okay. That all happens in prophase 1. Then we've got metaphase 1, which I will draw right here as M1. And that's when you have the lining up of the chromosomes, the homologous chromosomes, which have already crossed over during prophase 1. So I'm going to draw them lining up. I'm also going to try to show that they've already exchanged some genetic information. Remember that they can line up and orient randomly. So we could see 2 reds on the right side, or we could see 1 red on the right side, 1 red on the left side. It just depends on how they oriented themselves when they lined up during metaphase. And now I'm going to show that they've exchanged genetic information, that they've crossed over already in prophase 1. So they've exchanged some genetic information, increasing genetic variation, okay, which is an important idea. Okay, then after metaphase 1, we have what? PMAT A for anaphase 1, right? And that's when we have our, got your centrioles over here. Remember the centrioles where the spindle comes out, and then you have the spindle fiber, right, reaching out like this. Spindle fibers are reaching out. Okay, here we have the chromosomes. Okay, spindle fibers reaching out and separating the homologous chromosomes from each other. We still have replicated chromosomes there, but they are now, the homologous chromosomes are being separated. So then we've got PMAT, remember, T2, telophase 2, and this is when we have 2 new nuclei forming, right? 2 new nuclei are formed at the end of telophase. Oh, that should be telophase 1. Okay, 2 new nuclei. And we've got in one of the chromosomes, we've got how much information in each of the nuclei, how much information would there be? Should be haploid, right? Half the number of information, because now we've got 2 chromosomes, 2 chromosomes. Over here we had 4 chromosomes. Over here we had 4. Over here we also had 4. But now we have 2. Half the genetic information means haploid. Okay, and then cytokinesis occurs. There's something called a cleavage furrow that actually starts to form. Right here, a cleavage furrow is what it's called in telophase. And then cytokinesis occurs, and now we have the 2 new identical cells, right? With half the genetic information, haploid. All right. And now we're going to go through mitosis 2, which starts with those 2 haploid cells and then takes those replicated chromosomes and rips them apart, right? So we go through PMAT again, and this is exactly what happens in mitosis. So now we've got P2, M2, A2, and T2. This is exactly what happens in mitosis at this point. Crossing over does not occur again. It only occurs in prophase 1. Okay, so if we have prophase 2, we're just going to show these replicated chromosomes sort of floating around. The nuclear membrane already dissolved again. And remember, they're not lining up in prophase. They're not crossing over either. Then they do line up, right? In metaphase 2, they line up at what is called the metaphase plate. Okay. And then we have the spindle fibers coming out, right? Oh, by the way, spindle fibers are present here. That's actually what helps to line them up in the middle. Okay. Same thing over here in meiosis 1. The spindle fibers are connecting to the kinetochore, helping them line up. Okay. And you do have the spindle fibers, of course, over here in anaphase as well. The spindles, the centrioles. Okay. And here in anaphase 2, they are being separated. The sister chromatids are being separated, right, with the help of the spindle fibers. Okay. All right. So when we do have this separation that's occurring in anaphase 1 and anaphase 2, this is called disjunction. Okay? And when we have something called non-disjunction, that's when, and this is one of your statements, so it's important, non-disjunction is when we don't have a proper separation of either the homologous chromosomes in meiosis 1 or the replicated chromosomes and sister chromatids are not properly separated in meiosis 2. And so what does that cause to happen then? Well, it could cause for these cells in telophase 2 that form, or sorry, telophase 1 or telophase 2 that form, could have more chromosomes than they're supposed to have, okay? And so when this occurs, that's when what happens? This is when trisomy 21, for example, or Down syndrome can occur. So I'm going to show that process happening here. Let's see. Right here. Okay, if it happens in meiosis 1, this is what it looks like. We end up with extra chromosomes in meiosis 2. Okay. This is normal division, right? Over here. Normal division is the first example. This is what happens if non-disjunction happens, right? Non-disjunction is the improper separating of the chromosomes of either the homologous chromosomes in meiosis 1, as is happening right here, and we end up with how many over here instead of just 1, 2, 3, 4 chromosomes. We do end up with 6 chromosomes actually in meiosis 2, right over here. Or if non-disjunction occurs during anaphase 2 of meiosis 2, as is happening right here, right, we can also end up having problems in the gametes right here. So as you can see, though, all of the gametes are messed up over here, are not what they're supposed to be, right? This one has an extra chromosome right here. This one has one, these ones have one too little chromosomes right here. Over here, it's actually kind of interesting. If non-disjunction happens in anaphase 2 of meiosis 2, as is happening right here, right, we can still have this cell produce 2 viable gametes with a normal 1n or just n chromosome number. In the end, these 2 gametes are healthy, but these 2 are not, because this one has 1 less chromosome than what it's supposed to have. This one has 1 more, 1 extra of what it's supposed to have. So what that looks like on a karyogram is right here. So you can see this is a karyogram with the homologous chromosome set up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, and then here we have 21. This is why it's called trisomy 21, because we have 1 too many chromosomes here, and this is what can happen if non-disjunction happens and there's an improper separation during anaphase, right? So you can end up with what is called Down syndrome. This is how Down syndrome actually ends up happening. It's due to the improper separation of the chromosomes. And why do we care about this? Because it is something useful to know, but also it's in your statement. So non-disjunction can cause Down syndrome and other chromosomal abnormalities. So now we can highlight that one because we have covered it. And here we have the last one that we haven't covered. Studies showing age of parents influence chances of non-disjunction. So essentially, this statement is just about you knowing that as you get older, as humans age, the chances of non-disjunction occurring increase, okay? The chances of non-disjunction occurring and having a genetic disorder in your child, such as Down syndrome, can increase as you get older. Okay, and that just has to do with some of the mechanisms in the cells not working properly as you get older. Sort of some of your life functions just don't happen as easily or as efficiently and effectively as they used to due to the aging process. Okay, so that is all that I have for you for part 3 of 3.3. Meiosis. I hope it was helpful. Please let me know if it is helpful, because if I get good feedback on this, then I'll keep doing it. If not, I do not want to keep doing this. Thank you very much. Have a nice day.